So today is the last day of class. Yay, right? Yay. Yay. Anyway, yay for you. Sad for me, but that's how it always goes. So there's a bunch of business. We're going to finish up today with DNA binding proteins. I have a lot of material to cover, but I'll still probably have a little bit of time at the end if you want to ask any questions at that point. A couple of bits of business before we start. One is that evaluations are live right now. You should have gotten an email about it and a link. So please fill out evaluations. If we have time today, I'll definitely give you time in class to do that. You can access it from your phone and all that stuff. But, the bigger return we get on those, kind of the more complete information we get. Second thing is that regrades are due in my hand by Monday. After that, I just won't look at your exam. So please make sure that you are happy, or at least maybe if not happy, clear with how your exams are graded. And if there are any discrepancies, we absolutely want to take care of that. Let's see what else. Oh, yeah, there's a problem session next Monday, 12 to 2, in 1204 Herring Hall. A moderate-sized room, but it should hold everybody who wants to come. Let's see. And, of course, our final is next Wednesday at 6 to 8 p.m. Not my choice of a great time to take a final. I know many of you have 123. At least that's in the morning, so that's good. You'll have a nice gap in time to kind of rest up between those. If you ask me, study for my class. Wilson's class, who cares? No, just joking. Dave and I are good friends, so I can do that. But anyway, Dave's class is mathematical. Presumably you'll have done all your stuff that you need to know already. It's just algebra, haha. In any case, this class has lots of facts and things that you need to study. So if you're trying to figure out how to parse your time, well, obviously do what you think is best. But don't forget this class. I've had that happen in the past where I've had a final on the same day as 107B or A, and then nobody studies for mine and they do badly, which is sad for me. So I'm expecting you guys actually to do pretty well. Okay. I think that's it for announcements. Any questions? Oh, yeah, I will be posting the answers to set 4 today. I will also be posting, I'm trying to think if I posted the complete answers to set 3. If I didn't, I will. And also I will be posting sample final questions today or tomorrow. Okay, and I strongly suggest that you look through those in the same way that you get good clues about what's going to be on your exam from there. For the midterms, that's also true for the final questions. Of course, no answers will be given with them, so if you try and do them, that could be a topic of discussion next Monday. Okay. All right. Any questions before I get going? Okay, so I'm going to backtrack just a little bit through my lecture, because this is at the end when I was starting to fall apart. So let's talk about just kind of how proteins recognize DNA, and there are 3 things that I mentioned. I mentioned that they recognize the backbone. I mentioned that there are direct contacts between the bases, the edges of the bases, and the protein itself when it interrogates the grooves. And I also talked about how when proteins bind, they also deform the DNA, and that deformation is somewhat sequence-specific in terms of its energetics, and that can contribute to binding. So I'm just going to whiz through those to make sure that you got it all and give you an opportunity to ask any questions. So in terms of the DNA backbone, most of the interactions are charge-charge interactions. There's sort of a bulk charge interaction, which is to say that you have a basic binding face or a basic protein binding to DNA, negatively charged. That definitely gives them an advantage right off the bat. Then there's the usual lock and key fit of shape-specific interactions between the protein surface and the DNA. And the DNA has several topographic features that are really pretty prominent, one being the grooves, the major and minor grooves. And then there's the phosphodester backbone, which forms kind of a ridge that's negatively charged. So those are things that can be complementary to the surface of your DNA binding protein. For example, we know that the major groove is large enough to admit an alpha helix, and many DNA binding proteins, although not all, will stick a helix into the major groove. The minor groove in its normal state is too small to take a helix, but we're going to talk a little bit today about cases where proteins can pry open that groove and make it bigger. But in any case, mostly that would just be a small piece of protein going in there. Then, of course, after we kind of take care of the big stuff, there's the details of the hydrogen bonds, the phosphate backbone. Those are quite common. But nonetheless, you often will see basic residues and crystal structures that are next to the phosphate, but they're not clearly making any sort of actual contact. That could be still an ionic field effect. And then the last thing I mentioned, which is sort of something you probably won't learn about in most classes, is the counterion release. And really it comes down to the fact that if you look at DNA and you model it, and this has been borne out by experiment, is that DNA basically has a sheath of ions around it because it has such a high charge density. And that ion sheath has a very well-defined thickness, and the concentration of ions with it are basically independent of the external concentration. The binding energy is so strong that those ions are always there. They can be exchanged out, so they're mobile, okay, but they're not. They can never be depleted, at least unless you're like in a mass spec or something like that. So the amount of neutralization that actually happens. So first of all, one of the things that comes out of Manning's theory is that in spite of no matter what you do, for the most part, cations, monovalent cations can't really, and that's the majority of the ions in cells, can't completely neutralize the charge, which means even in a reasonable amount of salt, there's still the potential for an electric field from the DNA. Because really the neutralization of the DNA is not salt dependent, the question becomes why does salt disturb protein-DNA interactions? And I want to remind you that there are also unsatisfied charges and dipoles in the protein, and those certainly are sensitive to being screened out by the salt. Okay? So being a linear distribution of charge is a different situation than having point charges, which can be solvated. Okay. And then the last thing is that polyvalent cationic DNA binding proteins get a big advantage, okay, to binding to DNA because the valence of the ion determines the degree of neutralization, and the more neutralization you have, the lower the free energy. So a plus 10 protein, which there are a number of DNA binding proteins that are that basic, can neutralize almost all of the charge and get a big binding advantage just from that. And really, the overall driving force for that binding advantage is entropy. If you have a polyvalent ion, you can displace a lot more monovalent or divalent ions, and that helps things out. As I said, the other thing I want you to remember, and that's an entropic effect. DNA can never be completely desalted. People do this all the time, and what you're really doing is desalting the external solution, which gets rid of the majority of the cations. So it's not bad. It's just don't think that you're getting rid of all the salt there. Okay, I think I said everything I wanted to say about that. Then we get into details of actually protein contacting DNA. And just to remind you, in a normal A form or B form, if we're talking about DNA, it's B form. The DNA, there's only two places that a protein can figure out where the bases are, unless it unwraps the entire DNA strand, which is pretty unusual. There's some proteins that do a fair amount of unwrapping, but none of them do that and then interrogate the DNA bases in a sequence-specific fashion. Single-stranded DNA binding proteins do that, but not double-stranded DNA binding proteins. And so the two groups are where they can tell what base pairs are at a particular position. And that's because there are steric features, like the shape of this whole thing, okay? And then there are also hydrogen bond donors and acceptors, and each pattern of hydrogen bond donor acceptors is unique for each base pair type, both in the minor and in the major groups. Now, many years ago, people said, well, you have an acceptor, space, acceptor pattern here. That's less information than, say, here, where you have a acceptor, donor, acceptor, or if you flip these over, they're the same. And that's just simply not true, okay? Sterically, these are quite different. So really, AT and TA do look the same, even though they have the similar acceptor-donor pattern. Likewise with the GC base pair. Okay, and we just talked about that. Oh yeah, and one thing I wanted to remind you about is that the methyl group of thymine is a particularly important feature for proteins recognizing DNA. As I said, a number of DNA-binding proteins that recognize AT base pairs recognize it through the T-methyl. So it's a very special feature that proteins have evolved to learn how to recognize. All right, so how do they do this? Well, they do this by providing complementary hydrogen bond patterns on their surface to what's seen in the grooves. And in the grooves, okay, you definitely have, like I said, distinctive patterns depending on the bases. And so the typical thing might be to, say, have an arginine that has two nice hydrogen bond donors here, lining up beautifully with the hydrogen bond acceptors on guanine. In fact, this arginine interaction, either in the way that I've drawn it here or via these two guys, is the single most shared match between all proteins and how a specific amino acid will recognize a specific base. So in other cases, you think there might be a code, but there really isn't a straightforward code, but this is as close as it gets. Something like more than 10% of the time, guanine is recognized by an arginine doing something like this, okay, making this bidentate interaction. As I said, everything else is a lot more variable. And so really, with the most part, what you expect to see is you expect to see donors and acceptors matching up on the protein surface. Having said that, I definitely expect you to be able, okay, if I give you a base pair, I say, here's an AT base pair, and here's glutamine, show me how glutamine can recognize that AT base pair specifically, and not recognize any other base pair, I expect you to be able to draw that in either group. Okay, guaranteed there will be questions like that on the exam, without a doubt. Okay, so you're warned, and it's a substantial amount of credit, something in the order of 16 to 20 points will be that. Okay, because you're supposed to know your structures. This is how I get to testing. Okay, and what they mean. Okay, so this is not a GC base pair. These are just two different. These are two different examples of interactions, and this is a direct interaction with protein directly making hydrogen bonds to the base. This is another case where we have a cytosine that's being recognized by glutamate, which has the complementary hydrogen bond pattern, but this glutamate just can't reach the cytosine, and this interaction is actually mediated by a water molecule. Okay, these are hydrogen bond distances. So that turns out to be just as real as the protein touching that directly, and that's because DNA has a lot of hydration in the groups, and some of that hydration is fairly stable in the sense that it still exchanges in and out quickly, but it's stuck there at a high occupancy. And those you can think of as being really features of the DNA itself. And this is just showing that in a DNA-protein complex, what the hydration looks like is the same as what you see in the free DNA. Okay. And then we talked about how proteins achieve higher specificity. Again, if all the contacts were just simply independent of each other, it'd be hard to have a situation where changing one base pair in a recognition site would really have significant effects on binding, like more than tenfold. And so the way that proteins do that is they don't just simply recognize each base pair with one interaction, but they often will tie their interactions together, like in this hydrogen bond network in the restriction enzyme HINC2. Where we see specifically recognizing residues, glutamine in this case and asparagine, being tied together by this hydrogen bond bridge. And it turns out if you disrupt any one of these interactions, it's going to transmit that disruption through the hydrogen bond bridge by essentially increasing the mobility of these residues. And when mobility is increased, interaction strength goes down. So here's one way, is just simply tying this down and aiming it directly at that group. Here's another case where you have one residue that's making two interactions in the major groove right next to each other. And so those are definitely linked together because they're all through one residue. Okay, so that's called direct readout. Indirect readout is doing things where there's actually not, there's sequence specificity, but the specificity is not mediated by direct contacts. It's mediated by how easy it is to deform the DNA in a particular way that's sequence specific. So remember I said that proteins often, not always, but often deform the DNA when they bind. When they do that, the deformations are definitely going to cost some energy. Whenever you push something away from its lowest energy configuration, you have to pay for that. And it's the amount that's being paid for that really the protein is sensing. And this isn't as specific as a direct contact, all right? It isn't, but it can have substantial effects on specificity. So, for example, I told you about an example with phage 434, which is a repressor dimer that binds two arms of a symmetrical site. It's got inverted repeats, but in the middle of the inverted repeat, there's an asymmetric region that's not touched at all by the protein, but substitutions in there do, in fact, affect the binding constant. And shown here, in the middle, right in the middle of the bend that the protein makes when it binds to the DNA, it's between these two steps, the TPA base step. And the TPA base step, remember, is has one of the least strong stacking energies. It's one of the most deformable base steps. When you replace that with a less deformable base step, you lose 60-fold in binding. That looks kind of like specificity to me. It's not necessarily specificity for TPA. TPG would probably work just as well. But the point is that here's a pretty obvious example of where changing how easily the DNA is deformed without changing any of the specific contacts has pretty profound effects on binding. Okay. Any questions on that? I'm going to skip over this looping business because, I'm not going to skip over it in the sense, I want to talk specifically, this part I'm not going to talk about because it's really going back into the old world and almost nobody uses this anymore. This, though, I am going to talk about because it's something that comes up, which is if you have a protein and you're not sure if it loops, but you do know what its recognition sequence is, you can play with the distance between recognition sequences and ask what do they do for binding. So, for example, this protein has two DNA binding sites that are separated by some amount, presumably dozens, if not 100 plus hundreds of base pairs. And when the protein binds, it has two DNA binding phases on it, and so it can form this loop-like structure. Okay? And you can detect that by gel electrophoresis, because these guys move much more slowly through gel, than with the linear complex. However, if I now change the distance between these sites, and what I'm doing when I do that is I'm rotating the two sites with respect to each other. That's phasing. We did talk about that, and I still expect you to know about phasing. Okay? If I change the distance by, say, 5 base pairs, that's a half a turn of DNA. So now I've taken the two sites that are facing in some particular direction. I've essentially reversed the direction of one of those sites. Now if the protein is going to make a loop, it's also going to have to rotate and twist the DNA. That's quite hard. So I would expect to see, if there was a loop formed, a pretty profound effect on the DNA binding affinity. On the other hand, if loops weren't formed, then you wouldn't have that effect, and it wouldn't matter where the site was pointed. Okay? And then you can prove it again by adding another 5 base pairs to show it's not a distance effect, and you should restore binding, because now you've brought the site back to its original position relative to the old one, just spaced apart by 10 base pairs, which for a protein making a loop is not a big deal. Okay? If you have a couple hundred base pair loop and you change the distance by 10 base pairs, that really won't have much effect as long as the site is pointed in a particular direction. Any questions on that? So the other thing I want you to get out of the fact here is that you can use gels to kind of puzzle this stuff out and you get some very, very weird effects. I'm not going to give you something as subtle as this, because that's just too much. But I might ask you to, kind of look at some gel data and decide whether looping is going on or not. Okay, so that's where we left it off. Any questions? Yes?
Going back to the phasing of the
yes.
The last one, full phase.
Yeah, full turn.
That bend is still happening. Does that change? That changes the...
So this bend here is kind of, this is just showing that it can't touch because this is pointing in the opposite direction. This bend doesn't really occur permanently, right? There's nothing to hold it there. Right. Okay, so it's just showing. But now that this is now facing back towards the original site, like it is here, then it can interact in the appropriate way. Did I answer your question? Okay.
Also, when the tandem base pairs are added, it also depends on the sequence too, right? So what sequence you put in there?
Not much. No. Why? I mean, okay, you're right. There are slightly different helicities. Okay, but for the most part, 10 to 11 base pairs should take care of it. There's enough torsional flexibility that if you have a slightly, like the only time that would be really a problem is poly A, poly T. Once again, okay, because poly A, poly T is very much underwound, like 10 degrees underwound compared to other per base pair. So that would give you a not favorable situation. But just sticking most generic DNA sequences in there should work fine. Yeah, I mean, always when you start sticking sequences and things, you do have to think about what you're doing. I completely agree with you. And I certainly may ask you some questions on the exam that give you a DNA sequence, kind of like the first exam where I said, where are the points of bending here? Where are the parts that might be contributing to flexibility or not? Okay, and remember, it's all about stacking energies. Any other questions? Yeah?
Can you explain the band DC?
Yeah, sure. I did that last time, but I'll do it again. Okay, so this is the band shift gel right here. Oops, excuse me. Come on. All right. This is a gel and it's running in this direction, so this is moving the fastest. Okay, and the naked DNA typically will do that. You add a protein to it, and let's say you can form a It's possible that this won't loop just automatically. There might be an equilibrium between these two species, so you might see an open linear form and a closed loop form. The linear form will always run faster than the loop form. Okay, circles are the slowest. Remember we talked about supercoiling and all of that. Okay, circles are slow. So you might see an equilibrium mixture. You may not see B at all. Okay, it just depends on the particular protein. Okay. Now you keep adding protein. Now what happens is that you're now able to occupy both sites at the same time on one piece of DNA, and that will very quickly capture another piece of DNA because now you have twice the affinity. So at higher protein concentrations, you might see it shift up to the point where now you even have higher, slower moving species. And that could just be one of these bridged dimeric fragments. Keep adding protein, and now you're going to compete off the other piece of DNA. Now you're just going to have two proteins bound. That will actually run slower. So you can imagine as you increase protein concentration, it can be kind of confusing. You first start to see B and C. They go away and start forming D. And then you add more protein, you get E. That's not just building up a bigger and bigger complex. That's doing something different. Good question. Okay. All right, so let's move on to other aspects of specificity. Really, the reason I keep harping on specificity is when you think about it, a protein has to recognize its DNA sequence in really a soup of a lot of DNAs. And it has to do it in a really quick and efficient way. That's another question. But what has evolution done, to really increase specificity of DNA-binding proteins? Clearly, they have to recognize a lot of sites. And to some extent, this need really depends on how much DNA it has to sort through. E. coli has 4 million base pairs, so it has to, for a site to be unique in E. coli, it has to be unique within a 4 million base pair sequence. Humans 10 to the ninth base pairs. Okay, 1,000 times larger, basically. Some organisms are another 2 orders of magnitude larger than that. Maize has about 10 to the 11th base pairs. It's enormous. So, clearly, if the genome was random, which it is not, but if it was random, to create a site that would be absolutely unique by chance is going to depend on the size of the genome. So this is what I'm going to try and explain to you. So the question is, how large of a DNA site is required in a particular genome to be unique? Okay. So, for example, the E. coli genome has, and the statistics I'm giving you are not precise, okay? They are approximations, because the precise ones have messy formulae, which you're never going to use, because we're not going to test you on it in this class. If you want to study, genetic statistics, there's books on this sort of stuff. You can be more precise, but it's good enough to get my point across. So, as I said, we have 4 million base pair genome. So then the question is, in order for a site to be unique, and if you assume, which is not true, but that the E. coli genome is a random assortment of all the bases in equal amounts, okay, the probability of any particular sequence occurring, okay, is on the order of 4 to the minus n, where n is the length of that site, okay? So any given sequence of any length occurring randomly would be that, okay, 1 over 4 to the n. That's just the total number of possibilities. 4 to the n is the total number of possibilities of a DNA sequence of any length, okay, and then one of those would be 1 over that, right? Okay, so basically, if you kind of do the math, for E. coli, all right, you need to have at least 11 base pairs, okay, to be unique if it was all random, okay? So the site has to be at least 11 base pairs long. And kind of not surprisingly, if you look at DNA binding proteins from E. coli, typically they're dimers, and they typically recognize 4 to 7 base pairs per dimer. So that kind of fits in with our sort of general view. Now, for a human, things get bigger, okay, and you need greater than 15 base pairs to be recognized, okay? Now, that's, when you think about it, factor of 1,000 only gives you 4 extra nucleotides that stick on there. It doesn't seem so daunting. And we'll talk about, nonetheless, that it is challenging to recognize that site that big by a single protein. So here's sort of the problem. And it's not a complete problem. It's not something you're going to sit up nights worrying about, but it's a way of thinking about it. So if you have a globular protein, which is roughly spherical, okay, then essentially, it can cover a site that's the diameter of the sphere, call it, right? All right, so essentially, if you kind of do the math, CRO, which is a very small DNA binding protein from lambda, is an 8 kilodalton monomer. It contacts in a crystal structure of 4 to 5 base pairs per monomer. Okay, and that's about the right size, as it turns out. That's as big of a site as it can cover, given its size. To contact 15 base pairs, you need 9 times more protein. So 3 times more bases, you need 9 times more protein. Roughly, the size of the protein will go up as the square of the number of sites that you want to recognize. Now, that's in a very simple-minded situation, extended DNA in a spherical protein. So one of the solutions is to wrap the DNA around the protein surface, increasing the surface contact. Okay, and that's certainly seen quite a bit. All right. The second thing is, don't use a globular protein. Don't use a surface that's spherical. Use a surface that, say, comes in modules. So, for example, zinc fingers, which we will talk about later today, are small modules that recognize 3 to 4 base pairs each. And if you string them together, then they recognize that many base pairs. So they scale linearly with size. That's also quite common. The third thing to do, which is similar to the zinc finger approach, but instead of growing your protein, just simply multimerize proteins instead by non-covalent interactions, using dimers, trimers, and tetramers. And the thing nice about this sort of approach is that it not only increases the size without having to increase the size of your gene, you're just introducing a protein association, you also are going to improve affinity. So let's look at that a little bit more closely. So homomultimers, okay, that means multimers of the same type, are very common for DNA binding proteins. As I said, many DNA binding proteins, but not all, are dimers, okay? And typically they're twofold dimers. And remember, DNA has a bunch of twofold axes going through each single base pair or going through between each base step, all right? And those are going to perfectly match the symmetry of the DNA. So that makes sense. And if they do that, then they're going to bind the same sequences, right, but in a twofold orientation, and we call those things inverted repeat structures, or palindromes. So palindrome structures, sequences appear all over genomes. And we know that there are actually multiple reasons for that. That would be a great test question. I've never asked that one, but I think it might be that. So the thing is that one reason we see palindromes is that they form stem loop structures in RNA. Okay, and we know that those are important recognition sequences. They also form landing pads, if they're in double-stranded DNA, for dimeric molecules. So there's two reasons why you might expect to see palindromes in a sequence. Okay, so this helps because a small protein can now be made bigger for free, essentially, by just simply associating with itself. And it turns out that each individual protein doesn't have to discriminate as much in sequence, because now when you make the site bigger, you're really adding discrimination already. And the binding energies are basically going to be roughly additive. There'll be a caveat to that in a second. So here's kind of the basic example. Say I have a protein, it binds as a monomer, it binds to a 6-base pair sequence. Its delta G of binding, I'm just making this up, okay, it's minus 5.6 kcals per mole. And this, if you kind of do the math, gives you a KD of about 10 to the minus 4th molar, so 10 micromolar. 100 micromolar, which is very weak. Most DNA binding proteins that are real don't bind so weakly. Specificity-wise, this isn't so good because a 6-base pair site occurs about 1 in every 4,000 base pairs by random. Okay? So this protein has low affinity and low specificity. Now, if I just simply use nature's protein engineering, evolve a binding surface between these two, so now it forms a homodimer. Now things get much better. The delta G of binding is additive. Okay, so now I get twice the binding, but in terms of KD, that really ups the KD because those are multiplicative. And now you have something that's 100 nanomolar KD, which is actually quite respectable. 10 nanomolar KD. That's quite respectable. That's typical for a DNA binding protein. And the site occurs 1 in 20 million base pairs. Okay, so essentially, you get complete uniqueness, all right, for that. You get high affinity and high specificity for what? For evolving this little interface. Now, caveats. Obviously, if the way that these come together requires the DNA here to be distorted, then that's going to subtract from that binding affinity. Anything that you have to do to distort the DNA away from its normal state will cost. But certainly, maximally, you can expect additive binding. So that's pretty easy. Any questions? So that's why there's so many dimers. This turns out to be easier than evolving a whole much larger DNA binding protein. Gets even better, okay? So homodimers are one way of doing it. And as I said, many DNA binding proteins are homodimers, but also a number of them are heterodimers. And they're heterodimers that can be exchanged. In other words, if you looked inside of a cell, you wouldn't find just one type of dimer. You'd find multiple different types of dimers that are assembled. So let me give you an example of that. This is a simple one. So first of all, like homodimers, you have larger sites, you get better specificity, but these can also now be assembled in different combinations, where you can now have a few proteins recognizing a much larger number of sites than you would if they were just only being able to pair with one partner. Okay? And this also allows you a much greater way to fine-tune specificity. So, for example, I mentioned homeodomains some time ago. Those are DNA binding proteins that look like a lambda repressor. They're often dimers, and many, many times they're heterodimers. And in fact, those of you know about homeodomains, their role is to set up patterns in developing embryos. That's one of their major, that's actually how they were first identified. And in fact, they operate on a number of different promoters and turn them on at different times. And the way that's controlled is by controlling their heterodimerization. Okay? So how does that work kind of conceptually? I have two DNA binding proteins that can bind as a monomer in this case. All right? They bind to A, binds to A, B binds to B. And I can make homodimers of those. Okay? So if I just had this situation, I would have two proteins being able to recognize 4 sequences. However, I can also swap these, right? So now I can have an AB combination or a BA configuration. They're not the same, are they? Okay. And now this thing can recognize up to 6 sites. So having A and B in just one single site, I now get 6 combinations of sites it can recognize. If I had 3 proteins, it goes up to 24, and it really starts to grow quite hugely, okay, as I add more and more proteins. This is how you can have a relatively few number of transcription factors recognizing many, many sites. Okay, say a couple of hundred transcription factors that are heterodimeric can recognize thousands of promoters by using different combinations and controlling those dimerizations again by either phosphorylation or expression at the RNA level. Okay, so that's pretty cool. So multimerization is actually mostly seen in higher eukaryotes because you have a complicated set of sequences. You don't want to have a machine that's unique to each site. That would be too many genes. Okay, you instead would like to use the tools that you have in different combinations to achieve different specificities. Okay. Any questions about that? All right. So kind of shifting gears here. This is an idea sort of thing. And really the question it has become, and it started out as a very simple idea and it got complicated, and it's still complicated, which is how do proteins find their DNA sites in a reasonably speedy fashion? We know, okay, that as soon as you turn on expression of, say, a transcription factor, it very rapidly goes to its site. As quick as you can basically get it to be translated, it's found where it needs to go. And yet, how does it do that in a sea of really sites where it doesn't want to go? And in vitro experiments suggest that they find them very rapidly, okay, on the order of the diffusion limit. And I had this big thing here because when I first learned about this many years ago, it was said they did it faster than the diffusion limit, and that was a paradox. Okay, and it led rise to this whole idea of one-dimensional diffusion, which I'm going to tell you about. But really, this guy Steve Halford kind of really went crazy and spent a lot of time trying to prove that this was a ridiculous result. And so he has this nice paper that you can read if you want about that. So, again, one of the things you should always remember about science, is that it is not static. We don't just prove facts and move on. We often make theories that are not correct and later need to be corrected, and this is one example of them. So, a number of different studies have asked the question is, how do proteins recognize their DNA? And what's the summary of all of those studies really is what I'm going to tell you now, that there are really three fundamental mechanisms by which sites get recognized, and it really depends on, what context the whole process starts. So here I have this big thing, this big chromosome, okay, that has a sequence in it that some protein's going to bind. And we always imagine it kind of looks like this. How to do that? It's got a little guidance system on it, okay, that says, bring me here. Are there pheromones? No, there's nothing like that, okay. So that doesn't happen. That happens very little time, a few times. More than likely, it will land somewhere else, okay. So this is unlikely to find this site by a random 3D search quickly because it's just unlikely. I mean, it could happen. That would be three-dimensional diffusion, and that's relatively slow. More likely, when the protein lands, it's going to land there. It's going to bind non-specifically first. And that gets back to the whole business of non-specific binding. You would think if specificity were the utmost importance, that proteins would have evolved to have extremely high specificities. And there are certainly cases where there's a difference of, say, 3 or 4 orders of magnitude, maybe even 5 orders of magnitude, between specific binding and non-specific binding. But it could have been more than that. I mean, there are many proteins that bind ligands where the specificity is much, much higher than that. So why isn't it completely gone? Why isn't the non-specific DNA binding gone? Well, one reason is because then you're going to really have a long time to find your site. You're going to have to test each and every single base pair by touching it, coming off, touching it, coming off. That will take too long. So most proteins will do this. So there are really 3 different search mechanisms that proteins use, and they're really random. They're not directed. There's no pheromones, okay? There's no a guidance system. This has got to happen just with thermal energy. And so one of the things that it was definitely proposed as the first way that proteins do this is called sliding. And sliding is just simply going back and forth on the DNA, okay? It can do that. It's well known that proteins can slide along DNA relatively rapidly, okay, without dissociating. Okay. The question is, how long do they do that for? And that's really where the original problem was. The people who sort of talked about the 1D sliding as being how sites would be found overall envisioned that this would just stay stuck and slide around until it found its site. But that actually would also take a really long time, even in one dimension. And furthermore, we know that, say, in chromatin and actually even bacterial chromosomes are heavily decorated with proteins. They're going to have to dissociate at some point. And then somebody finally did some kinetics and found out, well, fact is, you can only slide around maybe 60 to 100 base pairs before you're going to fall off anyway. Okay? And so that's probably not likely. Because the binding the non-specific DNA, although specific DNA complex can have a very long lifetime, even on the order of minutes to hours, non-specific lifetimes are very, very short. So that's not so good. So what about something else? Well, there's also another thing called hopping and jumping. And what this is, basically, is that the protein slides along and then it jumps off, but because the electric field of the DNA is strong, it rebinds, but it diffuses farther than just 60 base pairs. So a hop or jump is really a larger distance, and a hop is a shorter distance. So what I mean by that, this is hopping, that's jumping. Okay, and eventually, ping. Okay? So this is kind of, combining all three things. Let's start that again. Whoops. Sorry, let me go back. Okay, we start here. We slide around a little bit, then we hop around. And of course, I'm not showing the diffusion away because this would take too long. And so these were hops. Just make sure those are hops. That's a jump. That's a jump. That's a jump. And then it slides around. Oops, I found my site. Okay. That's still quicker than three-dimensional diffusion would be. Yes?
So the DNA is compressed in such a way where the jump will allow the sequence to be found. You know what I mean?
So remember, chromatin's highly condensed. Okay, so the DNA isn't just strung out all over the cell. We envision it that way because maybe that's how you learn it, but it's really not true. Now, admittedly, it doesn't look like a lamp brush chromosome all the time either. That's only during cell division. But there's a lot of DNA organization, and I don't know if you're aware. There's been a whole project to map what's the chromosome structure like in a cell. And it finds out that there is quite a bit of structure, quite a bit of co-localization of regions of DNA that are co-regulated, for example, which makes this problem a lot simpler. There's also parts of DNA that always seem to be close to each other all the time. So there's some sort of chromosomal architecture, and that includes different parts of different chromosomes, not just within one chromosome. So there's a local organization, which is kind of the loops and so forth that you've learned about in chromatin, but then there's an overall structure maintained of at least some of the chromatin to keep certain parts in certain distances between certain parts within proximity to each other.
And it makes it easier.
Well, if they're co-regulated, right, then if you have factors that have to coalesce and control the same genes, you can imagine that if they're all in the same place, the diffusion times are much shorter, okay, and the control can be much more really driven by local concentration gradients rather than over an entire nucleus. All right. The last thing I want to tell you is called transfer. Transfer requires that the protein has at least 2 DNA binding surfaces, so this wouldn't be for every protein. And the idea is that these guys can, not only is the protein able to move, but the DNA is also not necessarily restricted to one place. I mean, yes, there's large-scale chromosome organization, but still within, say, 10 megabase loops, you still have quite a bit of mobility, and so something like this could happen. So this is where the DNA bounces, so the protein diffuses with the DNA, comes in proximity to another DNA, and either hops on or just transfers by actually bridging it. Okay? And again, we find our way to our site. Yeah. Yes. So the DNA is moving also, is what I'm trying to say. So that diffusion also has to be taken into account. It's not just simply the particle itself, but the DNA itself is moving around. And when there are 2 pieces of DNA come in proximity to each other, that's an opportunity for the protein to hop onto another piece of DNA. They could. They could. So looping proteins have that, for example. But okay, so let me just say, if they bridge the DNA, then they need 2 DNA binding surfaces. If it just happens to hop off and jump onto another one that happens to be close by, then it doesn't, right? Does that make sense? But I guess what I'm trying to say is that the DNA diffusion also plays into this. All right, so so everything I told you is likely to occur. This is the present view of how proteins bind their DNA. There's 3D diffusion, but it's inaccurate, okay? There's sliding, which only can be local. There's hopping and jumping, which can cover greater distances, but still can't cover, say, megabases. And then there's transfer, which is particularly efficient for proteins that don't dissociate from the DNA very easily. Okay? All right. So that's that. Any questions on that? All right. So now I'm going to switch gears and talk a little bit about techniques. This is an old lecture, actually. I haven't updated it much, but most of what I'm telling you still is useful. But almost all of it's been kind of, and I'll tell you the points where it has been overtaken by essentially one genomic technique, which is called ChIP-seq. So nonetheless, people still do these things in vitro. So the first 3 things I'm going to tell you about are in vitro sorts of experiments, and in vivo experiments would be ChIP-seq or ChIP-chip. So I definitely expect you to be able to explain these to me or interpret data about these. So let's talk about footprinting first. So here's the question. I have a piece of DNA, say it's a kb or so long, okay? And I know that my DNA binding site is localized within that particular piece of DNA, but I don't know where it is. And I don't know anything about it. But I have purified protein, and I know the site. So this is a technique that's used quite a bit called footprinting. It's still used a lot to really prove that you know where something binds to. Okay, and what is based on the idea is very simple, that you can cleave a piece of DNA up with either an enzyme, DNase, or hydroxyl radicals, which attack the DNA backbone and cause it to break. But if a protein is covering a piece of DNA, then those regions won't be as susceptible to cleavage. That's the basic idea. Footprint, maybe call it shadow, I don't know. Okay, so you start with a double-stranded piece of DNA, because we're talking about DNA here, and you label one end, either with a dye or with P32 in the good old days. Okay? And the reason you label one strand is that you're going to see cleavages on both strands, but trying to interpret a gel with cleavages on both strands is really impossible. Okay, and so you really have to look at each strand individually. You'll see why that is in a second. So if I just add my cleavage reagent at the right amount, so I have to play with this, then maybe I'm going to get it so that each piece of DNA gets cleaved only once, but it gets cleaved in different places depending on the piece of DNA. Okay? So I'm not cleaving one piece of DNA in all these places. I'm cleaving a bunch of different DNAs, each once at these different places. And so what I'll get is a bunch of fragments of the labeled piece, at least, that are different lengths. Okay? That's what will happen in a mixture. Then I run them out on a gel, and I get this nice thing called a ladder. This is the electrophoresis gel. This is the direction of electrophoresis. The small stuff's at the bottom, the big stuff's at the top. And since these are cleaving every base in the way that I'm drawing it, then there's a nice even interval. Okay? Now I add a protein, and that protein basically wipes out cleavage at these sites, but leaves it everywhere else. So now I have a gap in my gel right here, okay, where I don't have efficient cleavage within that. And the real key to getting these experiments to work well is that the protein has to sit there, be on there stably for the time of the experiment, number one. And two, you've got to make sure you have excess protein so that you don't have free DNA around that can contribute to a background signal. Okay? And so this is called a footprint, and there's the footprint. So now, based on the length of these fragments, because I can, these are also at single base resolution, I can use a sequencing gel to line them up. You guys probably don't know what those are anymore, but there were these things called sequencing gels way back in the day. In any case, I can find exactly where in my sequence my protein is binding to. This is very, very common. It's called footprinting. So let's look at some real data. This is from a paper that I gave you to read. If you want to learn all about how to study DNA in vitro, DNA binding proteins in vitro, this is the paper to read. They use about every single technique known to human beings to map the binding of Arg repressor, which is a pretty interesting protein, to the DNA. Arg repressor is a hexamer that wraps DNA around it almost like a nucleosome. Okay? And that's kind of interesting. So this is a DNase 1 footprint over here on the right. So shown here is a lane where you just add DNase 1 and you're getting a cleavage reaction. These guys are shown with increasing addition of protein to them. And then these are sequencing lanes, which cleave either at A or G or C at T, okay, that help you orient where you are in the sequence, okay? So this is a reaction that you do on the very same piece of DNA, and you can read the sequence off of this. It's not as clean, obviously, if you had all four lanes, but this is good enough. And so what you can see is you see that there are definitely cleavages going on when there's no other protein present besides DNase 1. And some sites in DNase 1 really likes to cut, and other sites not so much. So DNase 1 has some specificity for CNA residues. It also it has some structural specificity. This probably is a region of DNA that breathes particularly a lot. Okay? Looks like a bunch of A's and T's here, but that's clearly not enough to say that. All right. So now what you see is that when you add protein here, most of the bands stay the same, but a few bands get lighter, like that one, like these. Okay? Like these. So the regions that get lighter compared to the other one are going to be the places where the protein is sat. So from this picture, you know that the protein is actually binding to a number of different places. It's binding up here, you can see. It's binding here. It's binding here. And it's binding down here. Okay? You also might notice, well, actually, we'll look at the other gel. Never mind. All right, so let's go over here. This is hydroxyl radical footprinting. This is even better because hydroxyl radicals truly do cut at every single base pair. And shown here is a hydroxyl radical ladder Again, here the sequencing lanes over here. You can see you get a nice, beautiful single base resolution ladder of where this protein is binding. This is with no protein present. As soon as you start to add protein, you see shadows on the ladder. Okay, you see regions of diminished cleavage, and those are clearly, okay, where the protein is sitting. There's something else you might notice also. Does anyone else see anything different than what I told you? Is everything getting lighter as I add more protein? No. Are some things getting darker? Well, that's different, right? So in one case, you're having protection due to protein binding. In the other case, you're having hypersensitivity due to binding. Anyone have an idea of why that might be? How can the protein binding make it more sensitive, the backbone more sensitive to cleavage reagent? What maybe is it doing? Yeah.
Maybe it bends it in such a way that it exposes it more than a regular lipo that we see in it.
Absolutely. So if you open the DNF by bending it or unwinding it, you make the backbone more susceptible to cleavage. Absolutely. Okay, so these hypersensitive regions are telling you where the DNA structure is being distorted, even though it's not being protected. It's actually being unprotected. Okay? But it's clearly these are places where the DNA is being bent. And again, large repressor wraps this DNA around it. Something you might notice is that there's slightly more cleavage in between than there is in the control. Those are the regions on the outside. This is just the other strand, and you get the same sort of patterns. They don't line up because these DNAs don't exactly line up with each other. But you see the same sort of thing. You see some hypersensitive bands, particularly on this particular strand. Okay? So this is because the DNA is distorting it. So if you look at a hypersensitive cleavage pattern, you see hypersensitivity, you can infer that the DNA is somehow being distorted there, either unwound or bent. And in your homework, hopefully you have tried it, will notice that that is one feature that is in one of the homework problems. Okay. So any question about this? So what you learn about this is what regions get covered by the protein and what regions also get distorted. Now there are many, many variations on this. I wouldn't dare go into all of them, but if you want to learn about them, you can read about them in that paper that I told you about. So potassium permanganate actually detects DNA distortion quite well and cleaves like crazy in places where the DNA is bent. So that's actually used in a more kind of quantitative way. There's this thing called extension mapping, which is just a variation of what we talked about here. Rather than taking the labeled piece of DNA by itself and running it out on the gel, you have a primer and you extend the fragments to where the cleavage sites are, and then you map them that way. Why would you use something like that? You'd use something like that if you're trying to map something in a genome, okay, in a genome-sized piece of DNA, something too big to make a defined piece out of. But you know where it binds, and so you can add specific primers that'll probe the sizes of the fragments that you form in a very large context. That works. There's also this really cool thing called interference, which is a lot like what I'm telling you about, except here you do the treatments before you add the DNA binding protein. And then what you do is you look at the proteins that the DNA still binds to. Let me give you an example. So hydroxyl radical cleaves the backbone. And if it cleaves the backbone in the middle of a DNA binding site, the chances are the protein is not going to be able to recognize that DNA binding site anymore. So the way you do the experiment is you do the same thing. You add enough hydroxyl radical to cleave each fragment of DNA once, okay? And then you add the protein to it, and you separate the bound and free proteins from each other. Any ideas how you might do that? Sorry, bound and free DNAs. How would you maybe separate the bound and free DNAs? We talked about one method by which you can do that already. Sort of one way that's obvious from, say, 120L, and the other one is maybe obvious from what we talked about. Yes? Well, how would you get it to pellet?
You do it in like a gradient.
Yeah, you might be able to do that. Sounds too hard to me. Sorry. Yeah. Run it through a gel. Yeah, do a gel shift. Okay, and you have shifted bands and unshifted bands. You cut out the shifted bands and analyze those in the gel. Okay. Then what you're going to find is you're going to find wherever the modification that occurs that wipes out binding will be absent. So it looks just like a footprint, except in this case, wherever the protein was able to bind to, those cleavage products will be lacking. Okay, because the protein can't bind to the cleavages that occurred there. That's exactly how it works. You can also use an affinity column, too, which nobody sort of thought about. Put your protein on a column run the DNA over it, get it to stick, or pull down, which is really the same thing. Okay. So you can do this for any sort of modification. This is very nice because there's methylations that you can do. So you can add methylating agents. The N7 of purines is methylated. I think something I told you before is I want you to remember that. And some DNA binding proteins need to interrogate that amino group, the 7 amino group, to recognize for binding. And so if methylation at that site prevents binding, then you won't be able to see it. And what the methylation allows in a subsequent reaction to do a sequence-specific cleavage that gives you, again, different size fragments. Same thing, hydroxylamine deaminates cytosine to make it uracil. Okay? And so if a cytosine is important, then you can also further take that deaminated cytosine and actually induce a cleavage at that point. And so that cytosine happens to be important for modification. You won't see cleavage at that site in your interference experiment. Okay. The last way to sort of identify a site, which is kind of like way over the top in modern times, but there was a time when people really were interested to know where a protein bound is. So you can actually take your protein, modify it with a cleavage reagent that just creates hydroxyl radicals all over the place. But hydroxyl radicals actually don't diffuse very far. And so essentially if you take an EDTA-like molecule, attach it to your protein chemically, and then add iron and ascorbate, you'll generate hydroxyl radicals which only diffuse about 5 to 10 angstrom. So wherever your protein is bound and this hydroxyl radical spewing thing is located, it will cleave the DNA. You can use that to map sites that way. So it's an affinity reagent, if you will, for doing that. As I said, it's not used very much anymore with much, much better methods to do things like that. Okay. So that's gel-based techniques. Again, only useful really if you know what piece of DNA you're working with and you're trying to find out the details of what's important for binding. But all this can be abrogated, honestly, with crystal structures and mutational analysis. Another thing that comes up occasionally is you have a protein and you have no idea what it binds to. And maybe you're even so unlucky as you don't even know what organism it binds to, or you can't culture that organism, okay? Because I'm going to tell you about a method later that's better than this if you can grow the organism yourself. And so the way this works is that, first of all, you're able to make libraries of random DNA sequences. We've talked about this before, and I'll just remind you, you can synthesize DNA where during the coupling reactions you allow all 4 bases to be present, and you can get a more or less even distribution of bases at a bunch of sites in a row. So you make single strands that way, then you basically use a sequencing primer to add the other strand by DNA synthesis, and now you have a bunch of double-stranded oligonucleotides that all have the same length. They have priming sites on them for PCR primers, and they're potential substrates for a DNA binding protein. So now you take your DNA binding protein and you mix them with these guys. Most of the DNAs aren't going to bind. Okay? They're not, because they don't have the specific recognition sequence that this protein is looking for. But some of them will bind. Okay? And those are the ones you want to capture. And you can capture them in a number of different ways. You can use an antibody to the protein, okay, which is kind of the old way to do it. You can have an affinity tag on your protein, like a His tag or a maltose binding tag, and use affinity beads to capture it. Or you can run a band shift and cut out bands, okay? But in any case, what you want to do is capture the protein-bound DNAs. Those are the ones that are going to be at least enriched for DNA targets. You then take those, there aren't very many of them, PCR amplify them, run them out of the gel, and then you end up with a new pool of DNAs that should be enriched for proteins that bind to your DNA. Now, if you take these guys and you sequence them, say, do 100 sequences, you won't see anything. They'll look pretty much like your input DNA. And the reason for that is that, one, you have a very complex sample going in. The second reason is that the amount of enrichment you can hope to see really depends a lot on your experimental conditions. And what I'm going to tell you is that if you can achieve 1,000-fold enrichment in this experiment, you're doing really well. But here you might be starting with, depending on the size of your random region, say 10 to the 14th, 10 to the 13th molecules. Okay, so that means if the binding site is unique, it's 1 in those. Okay, so that's a very, very infrequent number. And now you get 3 orders of magnitude enrichment. Now you're in 1 in 10 to the 9th. Well, you're not going to see that by sequencing. Okay, you're not going to get enough sequences. You're not going to do 10 to the 9 sequences to identify that one excess that's there. So it's really a matter of having to run this cyclically. Now, having said that, my pal Dave Siegel over at Genome Center along with Ian Korf, came up with a script by which you could actually use high-throughput sequencing, which gives you millions of sequences back. And they could actually do an analysis that shows they could identify DNA binding sequences within a single round of enrichment. It's a little trick they call bind and seek. Scientists are the worst. Okay, but that doesn't always work, and it does, again, depend on how the work is done. So it uses bioinformatics to really sort through this complex mixture. If you're cheap, okay, or if you want to do this differently, you repeat this over and over again, say 5 or 6 cycles. And each time, if you enrich 1,000-fold, after 3 or 4 cycles, you should be down to the site. It usually takes more than that, because 1,000-fold enrichment is quite unlikely. Okay. Again, let's think about what gives you your background here. It's not only the infrequency of your site, there's also partial sites that bind okay, but not as well, and there's also just the non-specific binding capability of your protein. So the output from an old-style Cellex experiment would be a defined number of sequences, a few like this, and now from a normal kind of experiment, you'd expect hundreds of thousands to millions of sequences. But this is just to show that you don't need that many sequences to get some information. So this is a case where somebody took a binding region that I believe is about 35 random bases long, and they are probing this particular protein called FREAC4. And what they found is they got a bunch of sequences back. They sequence aligned them to find the best matching regions. And from this aligned set of sequences, they were able to deduce a binding sequence. So this is a table down here representing the bases that they found in each one of their binding sequences. So, for example, some sites are absolutely required for binding, like this T here, that C here, this A, these 2 A's here, pretty much that A here, that A. Those are all required for binding. So wherever there's a large letter here, you have a very, very strong preference for those sequences. Okay, essentially you don't get it. Every single thing that you recover are those sequences. But then there are other sites that have either no preference really whatsoever, like here. Okay, no statistically meaningful preference or very, very weak preferences where you have maybe an excess of T's or so forth, or you only get T and A, for example. All right, so those would be weaker sites. This site here, okay, is called the consensus site because it is really the most represented set of sequences at each position. If you feed the consensus site to the protein, it usually binds them really tightly. However, if you then take the sequence and go back and search genomes, you miss a lot of real sites, we now find. Okay, you miss them. Why is that? Why is the natural binding site often not the best site? Remember, there's a stock answer for all these evolutionary questions. It's not helpful, but it's an answer, which is... That's right, but why? Well, because it works. Okay, which is, again, not helpful. So why does it work? It works because maybe the protein doesn't need to bind so tightly to exert its activity. Maybe it, in fact, recognizes a lot of different promoters, or whatever, in this case, it's transcription factors, so I'm saying promoters, lots of different sequences of DNA. And so it's achieved kind of this parallel sets of specificity, recognizing different sites using different parts of it. Okay? The other reason is that, binding too tightly maybe mean it can't be properly regulated. Okay, so evolution has delivered, the appropriate binding sites for this protein, but they don't have to be better than that, I guess is what I'm trying to say. They don't have to be better than that. That's what's required. All right. So this is just kind of, in a way, trying to think about what's going on during the Cellex experiment. I've got 2 different plots here. Let's concentrate on this one. So what this is a sort of distribution, of binding energies. Okay, for a particular protein binding to a particular DNA. And this is a distribution in a population of molecules that you have. So, for example, let's just look at the n equals 0, which looks like a Gaussian distribution right here. This is, if you just went into your mixture of DNAs and you were able to measure the binding to each of ones to your protein that you're interested in, this is the distribution you'd kind of expect. There'd be some mean binding energy, which would be pretty low, and then you'd have, maybe a couple of kcals per mole on either side, although I don't know what the units of those are. So now you do one round of binding, and enrichment, and you reduce the size of your kind of non-specific peak, if you will, a bit, and now you have a little bump of specific stuff. Okay? So your overall population is not binding really all that well, but some of them are binding way better than before. I mean, you've got now, say, 1,000-fold enrichment. If you do the cycle again, now you really bring this up, and the peak of the binding affinity is going to continue to increase until you reach basically almost all of your molecules are kind of in the specific range of binding. Okay, and that's because each time you're enriching more and more for binding molecules and excluding fewer molecules, or more molecules that don't bind. Okay? Okay, and this is just basically saying the very same thing. This is from a real experiment. This is a theoretical experiment. And as you can see, again, the non-specific binders are over here. The diamonds are your first step. There's really no specific binders. And then by the time you get up to the fourth round, almost everybody is in the specific entity range. Okay. Any questions on that? It's a very useful technique. It's still used with new high-throughput sequencing methodologies. It can be done in parallel with a number of different binding proteins. It's pretty cool. Now, one bit of technology I'm going to tell you about that's really old, but still worth talking about because I think it's going to come back. Not for this particular methodology, but just in general. These so-called DNA binding microarrays. They still teach you guys about these. They were really hot from like 2000 to 2010, and then sequencing just killed them. So what a DNA binding microarray is made possible by is some really cool chemistry, whereby which on a silicon chip you can build up a piece of DNA of any sequence you want, and you can localize it in a very, very small part of the chip. And you can do that because there's laser-addressed synthesis. Without going through all of DNA synthesis involves rounds of coupling, deprotection, and then coupling again. Because you don't want to add reagents and have your chain kind of grow randomly. So each time you add a reagent, you want to stop at just a single step, a single base added, and that's done by protecting the end of the base that you've added on. And then once you've done the coupling there, you wash away everything, you deprotect those ends. Now they can act as new growing ends. And so if you could use a laser to deprotect them, which is exactly what they do, then you can deprotect certain regions and then flow your binding reagent, say for C, over the whole region, but only regions that have been specifically deprotected will take on a C. You wash away the C reagent, you now do the same in places you want to have A, G, and T. Okay? And so it allows you, as I said, in any part of the chip to really make a dot of a couple of microns, 10 microns wide of a specific sequence. And it can be any sequence you want because you can program a computer to do all of this. It takes a while. These guys are expensive to make. I don't know how much they cost now, but it used to be a couple of $1,000 to get a few of these made. And so this is just showing a real picture of a microarray that has different sequences on it where people have added primers that have fluorescent tags on them. You can see for the most part, every place has a dot, but there are still a few places where the chemistry didn't work and you didn't get something. Okay, that's why there are blank spots there. Then you can take these primers and extend them with fluorescent nucleotides, which gives you some color. Okay, and that's why some of these have been shown to have color. And then what you can do is add a protein with a fluorescent marker, okay, that's different from all the colors I've told you about, and you can visualize those proteins bound to the DNA. And you use the intensity of your extensions here to know what your DNA density is, because different DNA sequences are made with different efficiencies, so they have to be normalized. So some of these bright dots might be because they bind really tightly. The other bright dot might be because that particular area, the synthesis went very well, and you got a whole ton of stuff. So once you normalize that, then you can find out exactly what sequence your stuff bound to, because there's a bright dot at a particular coordinate, and you know what you programmed in to put there. So that's quite useful. Let me show you an example where that might be useful in an experiment. So remember I told you about zinc fingers? Well, zinc fingers recognize 3 different base pairs. Okay, they're a module, and I'm going to show you a picture of zinc fingers in a little bit. But these modules, okay, come from nature originally, but nature didn't recognize all the 64 triplets. It only recognized about 17. And so somebody, before we heard about CRISPR killed everything. It was great. Just killed all these technologies. But zinc fingers are still useful, as Dave Siegel will tell you, because he's deep into them. But anyway, there was a whole protein engineering effort to generate zinc fingers that recognize different sequences. And this involved artificial evolution, where you start with a normal zinc finger protein. You take a specific finger type, you basically subject it to mutagenesis to generate a population of mutants, and you can use affinity techniques to purify out the proteins along with their DNA that encodes for them by displaying them on the outside of bacteriophages. This is very fun, definitely, again, kind of dated technology. And you essentially get molecules using affinity methods that bind better to a particular sequence. And so this is a way of analyzing what comes out of those experiments, so you can kind of see what's going on in this in vitro evolution sort of thing. So, for example, there was no zinc finger for the sequence GCG, but there's a bunch of sequences that recognize GGG. zinc fingers like to recognize Gs for reasons we'll see in a second. So after they did one round of mutagenesis and selection, and what they're shooting for is GCG, okay, TAG was the initial, in this case, the initial specificity. So this is the spot. But when they do their second round of selection, trying to find things that bind GCG, they find that this new finger that they've got, or new population of fingers, binds a lot of different sequences. Okay? But then they go to round 3, and now they're starting to see a greater increase in affinity for the ones that they're looking for. And finally, by round 4, they've got a molecule that recognizes both GTG and GCG pretty nicely. Again, over here, TCC, this has no G's in it at all. So the first round, they basically got nothing. The second round, they got a very, very non-specific but tight-binding finger. By round 3, they were starting to recognize things that not only bound TCC as not the best one, but actually other ones as well. And so this is a way of really interrogating the specificity of a design molecule all at once. Yes, sir?
What are they doing in each round?
What they're doing is they're subjecting their pool of mutants to mutagenesis, okay, and then running them over an affinity column that is designed to be the sequence that they're looking for. And these guys are packaged into bacteriophages, and so on the outside of the bacteriophage is where this DNA-binding protein is living. And so the bacteriophage will be stuck to the column if it binds to that DNA. And if it doesn't, it flows through. So what you're doing is essentially an artificial evolution. You're introducing genetic variation. You're using a selection to pull out the ones that have the properties that you want. And then you're taking that whole mixture and interrogating basically a chip with that whole mixture of proteins. Does that make sense? Okay. All right. And so I'm not going to go into this. These are called logos. You're probably used to seeing them. They're ways of kind of saying how good does this bind to particular positions. And it's just showing that they were able to get, actually using this methodology, some pretty convincing specificity changes, starting with TGG and ending up with these other ones. Some of them didn't work very well. Some of them didn't work well. Some of them worked really well. Okay. The last thing is really the way things are done now. This is called CHIP. And CHIP stands for chromatin immunoprecipitation. Okay, it doesn't even really have to be immunoprecipitation. But basically what you do is you allow a protein in the context of an actual organism to find its DNA site. You then nail it there with a chemical cross-linking agent, like formaldehyde. You then fragment the DNA into pieces, and then pull out your protein bound to its DNA fragment using a specific antibody. That's the immunoprecipitation part. Then you have protein bound to DNA, you reverse the cross-link, amplify the DNA, and then somehow figure out what the sequence of that thing is and associate it with the genome sequence. So this is what you do when you have a protein, you know what organism it's in, or a gene, and you want to know where it's binding in the organism. Okay? So this is the workflow. So you start with your cells, okay, they are expressing your protein, tagged or untagged. You then add a cross-linking agent, like I said, the formaldehyde for a while, and that will trap the protein-DNA complex where it is. Too much cross-linking is a problem, then you pull down all sorts of garbage. Too little cross-linking, you don't get a signal. So there's some fooling around that happens. Then you lyse the cells, and now you have a bunch of chromatin, which is too big to do anything with. Everything is linked together in the chromatin, right? Not only because of the cross-linking, because you have a billion base pairs in a chromosome, or a couple 100 million base pairs in a chromosome that's going to drag everything with it. So you need to shatter this thing into fragments, but our good old friends sonicators are very good at doing that. And so by using sonication, you can break this down into, because it's still bound to nucleosomes, remember these are nailed on there, much larger fragments, but you end up with much smaller fragments, say in the order of a half a kb to a kb. Okay? And then those guys can be pulled down with specific antibodies, and presumably some of these fragments have your protein attached to them. Okay? Then you reverse the cross-link, which is pretty easy with formaldehyde. It's just a pH jump. And now you have these fragments, which then you can do whatever you want with. You can take them and PCR amplify them to look for specific sequences. You can do qPCR to figure out what the occupancy is. You can use a microarray, which is just a hybridization array like the ones I was telling you about before with the zinc fingers. Or you can sequence them by Sanger. But nobody really does that now. What everybody does is they do high-throughput sequencing. Because it's just so much easier and cheaper. You can run 10 to 50 high-throughput sequences in one MiSeq run, which is pretty nice. And then what you get here is you get the numbers of counts of specific sequences you brought down. So what you're doing is you're counting these guys, and wherever you have lots of counts, presumably that's where your protein bound. And this is actually the binding profiles along a particular stretch of chromosome, in a particular chromosomal region, of different proteins that are involved in a transcription activation complex. And I think you can pretty much see that most of the action here seems to be in this region and maybe here as well. Okay, so this is used to map promoter occupancy by different proteins. Yes? Well, RNA binding proteins, that doesn't help you here, right? Because an RNA binding protein, you could do an RNA-seq type of experiment. I shouldn't call it a chRNA-seq. I guess I'll call it that. It's not chromatin immunoprecipitation. That would be mRNA immunoprecipitation. And yeah, people do that. Okay? So what you would do there is you would basically take your cell, you cross-link it just like you did before. You digest away the DNA, okay, with DNase, which you can do. You then use an antibody to capture all of your RNA protein complexes. You then reverse transcribe those guys and submit them for high throughput sequencing. Interesting enough, I haven't seen that done as much, which is interesting. I think the RNA transcriptome is a lot more complicated, so it's probably a little bit more difficult to get good signal. And you don't have the fragmentation situation like you do with chromatin. Single-stranded RNA is actually hard to fragment. It's pretty flexible. Okay. Any questions? So that's what you do. So we have different methods depending on what you know. You've got footprinting methods if you have defined DNA and a defined protein in a purified environment. You've got CellX, again, if you have a purified protein, but you don't know what DNA it binds to. And then you have ChIP-seq, really, to really find out where in the chromosome your protein is binding and under what conditions. Because obviously you can vary conditions, stimulate the protein, and stimulate the cells in different ways and get lots and lots of cool information. So this is very cool. It's very heavy on the computation side, but really cost-wise, one of these experiments can now be done for $10,000, which is pretty good, from start to finish. Okay. Right, I want to wrap up with pictures because I think it's a good way to leave this class. Because really the class is about pictures. And what I want to do is I want to show you some pictures of some common DNA binding motifs. And I'm just going to tell you that for the most part, I'm not going to talk about tau proteins actually today because we don't have time for it. I usually end up with that. These are 3 common types of DNA binding proteins. There are a number more, and they have different sorts of things that I would like to tell you about them. But really, what I want you to get from these is basic principles, not the specifics so much. Okay? If I ask you anything about these guys, it'll be something that I hope is particularly obvious. All right, so these are modules, meaning that they're found all over the place, appended to different proteins that do different things. The one commonality is that they all bind to DNA. Although some zinc fingers actually don't bind DNA. Some of them bind RNA, which is interesting. Some of them even bind single-stranded RNA, which is even more interesting. And some of them are actually involved in protein-protein interactions and have nothing to do with the protein. So there you go. Leucine zippers, if they're basic region, they all bind DNA. Okay, if they don't, then they're coils, and we know those are all over the place. And the helix-turn helix proteins, again, most of them are DNA binding proteins, but a few of them are involved in protein-protein interactions. So let's start with the HTH, or helix-turn helix motif. Very, very common in both bacteria and in higher eukaryotes. And really, it's exactly what it says it is. It's a helix, a turn, and then another helix. This helix here, okay, is stuck in the major groove of the DNA. This helix is packed against the backbone, and this turn is always located in pretty much the same place. And so what I'm going to tell you is that essentially this is bioinformatically easy to recognize because this turn, there's a glycine and another set of residues here that are pretty easy to recognize, and then there are almost always basic residues on this part and this part that coincide with a particular part, and we'll see why that is. So the basic idea is that this guy supplies backbone binding, this one supplies both base-specific and backbone binding as well. And this turn is just a matter of putting these guys in the right position with respect to each other. Okay? Examples of this would be repressor proteins. In E. coli, many, many, many of them are helix-turn helix proteins. Okay, related to this is the homeodomain protein. The homeodomain protein is the helix-turn helix protein, but it has an extra helix in back of it. By the way, can I just back up here? All you need, in some cases, some of the helix-turn helix proteins are almost this small, or some of the helix-turn helix appendages, domains, are almost this small, 70 or 80 amino acids. Crow repressor is basically this with a little bit of extra stuff. Okay? So this is really all you need. And there's a little mini hydrophobic core in here. Now, homeodomains are definitely more common in higher eukaryotes. And as I said, they're a helix-turn-helix motif with the helix behind it, and between these three helices that make what is called a three-helix bundle is a hydrophobic core. So this has a real hydrophobic core, unlike the other things that I was telling you about. And the way that they bind is slightly different. This backbone binding helix is offset compared to the one I just showed you. The other one I showed you would cross right about here, and this one has taken its place. But more or less sequence-wise, they are quite similar. Now, one thing about homeodomains that's different from other helix-turn-helix proteins is they all have this N-terminal flexible arm. That's what it always says in the papers, okay? And this N-terminal flexible arm is involved in protein DNA binding. So let's have a look at the homeodomain up close, turn it around, and see what's going on. Let's start with this helix here. This is called the recognition helix, okay? And I want you to know that because it's very common, all right? And a recognition helix sticks into the major groove, like I said, and interrogates both the bases and the backbone. So shown in here in blue are side chains that are making specific base contacts, which are a little bit hard to see here, but believe me, they're there. They're actually touching the bases and important with specificity, either directly or through water-mediated hydrogen bonds. So these are the specificity really determining residues. And yet they're not the only ones, because you can change these. And typically what happens is that you relax specificity. Occasionally you get specificity changes, and more often than not you just kind of wreck the whole thing. This kind of goes back to the idea that you've evolved very particular specificity that involves a whole set of residues. Now, on the outside of these guys, flanking these specificity-determining residues, which are all faced into the DNA, so therefore, if I gave you, say, a helical wheel, you should be able to show me, for example, where you'd expect specificity-determining residues to be on, what positions they might be in. So that's, again, something where I can integrate the idea of helical structure into DNA protein recognition. But on the outer edges of this, there are these red residues, which are arginines, glutamines, and they're all making backbone contacts. So this part involves specific DNA protein contacts. This part on the outer edge of it really is where the non-specific stuff, which makes hydrogen bonds and salt bridges to the backbone on either side of this major groove. And then the first helix that I was telling you about, of the helix-turn-helix motif, has actually an arginine that makes also backbone contacts. And so this part of that first helix of the helix-turn-helix motif is making backbone contacts right here. Okay? All right. And that third helix is just, as I said there, to stabilize this whole thing by forming a hydrophobic core. Now, the N-terminal part is reaching around to the backside. This is the major groove. This is the minor groove. Remember I told you the minor groove is only big enough to admit a peptide? Well, here's the perfect example. So this little N-terminal tail is reaching around back and is making two sequence-specific contacts, this residue and this residue, with the minor groove. Okay? And this guy actually recognizes two base pairs, and this one recognizes one. Okay? So you kind of have, this one almost tells all the story, really. You've got a helix in the major groove and a beta strand in the minor groove. Okay, now most HTH proteins, well, I shouldn't say most, many are not monomers. Homeodomains can either be monomers or dimers, heterodimers or homodimers. But your typical bacterial HTH protein is a dimer. So this is the case of what I was telling you about. Here are two subunits that are identical to each other. They're related by this twofold axis of symmetry. And in fact, that's the same twofold axis of symmetry that goes through the DNA that it recognizes. This is just a side view. Here's the two recognition helices stuck into the major groove. Here are the two non-specific binding helices touching the backbone. So when Crow-repressor, which was the first DNA binding protein structure ever solved, was solved. It was solved without DNA, but it was in its dimeric arrangement. And the first thing that Dale Tronroot, who worked on that structure, noticed is that there are these two helices that were spaced apart perfectly to fit into two adjacent major grooves. Okay? Two major grooves, which is roughly about 37 or 38 angstrom apart. Okay? And so he hypothesized this model based on that, and in fact it was correct. Okay, it was correct. So later on structures came out, and pretty much exactly what they thought, including this little bit of bending that occurs at the corners in order to maximize the protein-DNA interaction. So in this case, there's no big bend in the middle, like in the other HTH proteins I told you about. The bending here occurs at the edges. Okay. Any questions on that? So as I said, HTH proteins are all over the place. This is our friend Crow, and lambda repressor actually bound at the same time. This is a homeodomain protein bound by itself. This is lac repressor. Remember lac repressor? You all learned about that, I hope, in 101. Kind of the lac operon. So lac repressor is an HTH protein, but it's atypical. It's atypical because it is, in fact, a dimer. It's actually a tetramer. Here's the lac binding region over here that controls DNA binding. Here are the HTH domains. they're bound in two major grooves, okay, that are apart from each other. But this is the funny part. Right in the middle is another helix, and that is jammed right into the minor groove. So unlike most helix-term helix proteins that either keep the DNA straight or bend it towards them, this one actually bends it away from them. Here, because Y has got two alpha helices shoved into the minor groove, that opens up the minor groove quite a bit. And that's going to cause the DNA to bend away from it. This is a so-called pure R family, and this is very specific to that family's binding. These helices in the minor groove that opened it up, and then the sequence-specific recognition is mostly happening out here. So it still binds and inverted repeats as the bend is away from the protein, not towards it. Here's an endonuclease, which is very complicated, but it has, in the end, one of its major DNA recognition portions involved the helix-turn helix, which isn't very visible. It's right over here. This is gamma-delta resolvase, another protein that also bends the DNA away from it. This is even more interesting, which is, again, it has two helices that are slid into the minor grooves, but rather than being jammed in like this, if the minor groove were here and we're jammed in like this, it flanks them, kind of like a leucine zipper. And then the helix-turn helix guys are at the end of this, and they're contacting specific recognition regions on this side. And all the chemistry that's going to go on is here. Gamma-delta resolvases or a type of topoisomerase. And then PIN recombinase, which is another DNA binding protein that causes recombination, has a helix-turn helix part of it. And again, it's kind of like a homeodomain. It's got some non-specific, or non-specific, non-major group interactions being caused by these tails. Okay, so that's HTH proteins. Any questions? Sorry to go through all the way here without a break, but I just want to get through this stuff. Okay, so the next protein I want to tell you about is this basic region leucine zipper protein, or BZIPs. Okay? And BZIPs look like a pair of chopsticks picking up DNA. Okay? We saw this early on in the quarter. These are two different views. This is looking down the helix axis of the DNA. This is looking from the side. Here's the coil-coil region where the leucine heptads are, and you can see how they're wrapped around each other. And here are the basic regions. Okay, so the basic regions is you see the coil-coil motif in the sequence, but then you have all these arginines and lysines lined up. And that's what really signifies a BL-HLH protein. So again, here's the leucine heptad, and all those leucines are spaced 7 apart, and you can see they're faced into the core of that bit. This is what a basic region looks like, stuck into the major groove. I didn't show both of them, I just showed one. And this is very interesting. So here are the arginines. There are 5 arginines here. Okay, that's a lot of arginines to stick into a small part. And basically, when DNA isn't around, that part is so positively charged that it flies apart. It's completely unstructured. So in these BZIP proteins, when the DNA is not there, the leucine zipper part is associated, okay? But the basic region is just flapping in the breeze. It's just a random coil. It's only when it encounters a DNA groove with a high density of negative charge can it assemble into this. And what you can see is that all of these basic residues are making contacts with the backbone. The only specific contact here is this residue here. All right? And let's test your knowledge, okay? So this is anyone see what base is that? It's a base. Oh, sorry. You guys need to tell me when you can't see stuff. I can't see behind me. Okay, so let's do it again. What base is this? Not this amino acid. What base is this? Cytosine. Okay, what's that thing? That's a 5-methyl. So that there has to be a thymine. Okay? That's a T. And therefore, this is an oxygen because it's red. So that must be receiving a hydrogen bond from this guy. Can anyone see what this one is? Okay? Is it pyrimidine or a purine? How many rings are there? 1. It's a pyrimidine. Okay? And that's a nitrogen, so that must be what? That must be what? Folks, better know those bases. You're going to be hating it. I guarantee you'll be hating this exam if you do not know your base pairs. Forewarned. Okay? So, what is it? Cytosine, right? Because thymine's here. It's got an oxygen. The only other guy that's a pyrimidine that has a nitrogen off of the ring is cytosine. So this is a donor, a acceptor, and this is a donor. What is this amino acid? What's that amino acid? We got a donor and a acceptor. Asparagine, correct. Why? It's like a beta carbon and a carboxylate, so that's aspartate or asparagine. And in this case, it's got a donor and an acceptor, so it must be asparagine. So asparagine, so it recognizes TPC base step, because these guys are in the same strand. TPC. That's it. 2 bases. Now, remember, these guys are zippers, okay? So if we go back and look at this, whoops. There's some distance between those, and it turns out that that distance is 4 base pairs, okay? So these two guys are actually 6 base pairs apart, the two TCs they recognize. That means they're on opposite sides of the helix, which of course makes sense, right? And so what's the recognition of the sequence of this guy on one strand? Well, it's TC on one strand, but it's 2 different strands, the 2 complementary strands. This guy binds a palindrome. Why? Because it's a dimer. So it's GA blank, 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 4 blanks, TC. That's a specificity. Okay? Now you may say, that's not really very specific. You're right, this is not very specific. These guys tend not to bind DNA all by themselves. They tend to be part of bigger complexes. B-zip proteins you might have heard of are Phos and Jun. Okay, they form a heterodimer. But MYC is another one that forms a homodimer and a heterodimer. Okay, they bind like this. Okay. Any questions on those guys? So I definitely expect you to kind of be able to look at something like this. I'm not going to show you these kinds of pictures because they're too fancy, too hard to print, but to really get an idea of how the DNA binding residues are distributed around there. Now, variations on a theme, okay, are these helix loop helix proteins. They kind of look like B-zip proteins, except they've got a leucine zipper region, which is again identifiable by its sequences. But it has a basic region, which you can again identify by the sequences. But there's a bigger bunch of intervening stuff in between, and that's because there's a dimerization region. And the basic region aren't part of the same helix. They're 2 separate helices, and these helices are connected by what looks like an unstructured loop. It's actually quite structured. It's just not regular structure. So it's a helix loop helix protein. Again, if we look at the zipper region here are the heptads. Not everybody's a leucine in this case. This is a methionine. This is a histidine. You often will see this in transcription factors where it's not all leucines. That means that this probably dissociates. It's probably not as tightly bound. This probably heterodimerizes, in fact. Okay, and it does. Here's the loop region, and here's the second helix, and here are the basic regions. And these are all the residues that are involved in DNA binding. And again, most of them, as it turns out, are basic. And the same thing goes, I'm not going to go to the details of this one, there's only 1 really, or 2 specificity determining residues. Only 2 to 3 bases are recognized by that basic region. All the rest is non-specific binding. Okay, any questions on basic helix proteins? Yeah?
Is the helix loop helix all just a single protein?
No, it's a dimer. Yeah, these are both dimers. Okay, so this is one chain, and this is the other. And just to be sure, it's also true for the leucine zippers. These guys all bind as dimers. And so the zipper part is the dimerization part. Okay, zinc fingers. I promised I'd tell you about them. So these are zinc finger proteins. This zinc finger protein is called ZIF268. It's actually a synthetic DNA binding protein, but that's okay. It has 3 modules. They're so-called zinc finger modules. And so I'm going to zero in on this particular module right here. Okay, they're called zinc fingers because they bind zinc. Okay, and the zinc is bound in a stereotypical fashion. The ligands are either histidines or cysteine, and in fact, the combination of ligands that you can have, which could be anywhere from his4 to cis4, and all the combinations in between, okay, are really kind of signatures of particular subgroups of zinc fingers. So the one I'm showing you now is called the so-called C2H2 cis2 his2 zinc finger. Okay, zinc likes tetrahedral coordination, so that's a nice tetrahedral coordination site over here. This actually really, with the exception of this interaction here, has nothing to do with DNA binding. The function of the zinc here is simply a stabilization. And this has been proven by showing that you can actually take out these residues and repack them with a hydrophobic core, and this still works. It's not as stable, but it still works. Okay, so this is structural. It's an organizing factor, and metals are really good for doing this, especially for small chunks of protein. However, as I said, there is this nice hydrogen bond with the backbone, which I'm sure contributes to DNA binding. The other thing that's fairly about this particular class of proteins is a highly conserved arginine residue, always at this position, and it's always making this kind of backbone contact. So this particular class, C2H2, always has this particular arginine, again, within a very obvious sequence relationship to the rest of these. In fact, these guys are easily recognized bioinformatically by the spacings between their cysteines and histidines. Okay, and that arginine there. So you can bind the genome for these very easily. like the leucine zippers, again, looking for basic regions in their case, and a zipper motif. Okay, so the way these start is a beta strand that has this loop that has a ligating residue on it, and it comes back to make another beta strand. These guys are paired together to make a very small beta sheet. Then at the end of this strand, there's a loop that comes around into this helix. This is so-called specificity determining loop, okay, in zinc fingers. And all the specificity determination for the triplet are right here, okay? And then it goes up into a helix and then off into the next module. These modules are about 60 or so amino acids long. Okay, let's zoom in on the DNA binding region, and we're going to start testing you on your bases again. So, as I said, zinc fingers recognize triplets, tandem triplets. They're all next to each other. Okay? And so let's start with this one. What side chain is this, folks? Even though I don't have the atom colors, just the shape of it should really suggest which side chain it is. Okay, it's long. Look at that. 1, 2, 3, 4, 5, 6. That's pretty long. It's only one side chain that long. Arginine. It's arginine. Okay? And who is this arginine interacting with? What base is this? Guanine, right? Okay? If you can't tell guanine, folks, you need to do some work. Okay? All right? And the reason is that this guy it's a purine. It's got an oxygen and a nitrogen here. So, first of all, what groove of the DNA is this guy interrogating that guanine with? Major. Why? Because this is the top and it's faced into the major groove. So this is a canonical arginine-guanine interaction. Bidentate hydrogen bonds perfectly spaced, just like in arginine donors and guanine acceptors. So that's 1G of the sequence that this guy recognizes. Okay? Let's go up to the next one. Now, this is a little bit hard to see, I admit. Okay, so you're going to have to bear with me. This is the middle base, actually, of the recognition sequence. This other C over here is actually part of the GC base pair that's below it. And this is actually on the other strand. This is a C. I'm going to tell you that because it's hard to see. Okay? And this is a hydrogen bond interaction. What residue is this likely to be? Okay, so is this acting as a donor or an acceptor? Acceptor, right? Because this is the donor over here. So it's two possibilities. It's either aspartic acid or asparagine. Turns out it's aspartate. Okay? So even with a negative charge, it's still good. So that's a GC base pair below it. This is on the other strand. So it's GG. We have that specificity. And now the last one is this histidine. Okay? Oh, I should have asked you. That there's histidine. Okay? Now this is funny because the histidine is aimed at what atom is this? We've talked about this atom a lot. Well, anyone know what that is? A purine? It's N7. Okay? This is N9. Okay? What base is this? I told you it's a purine, but what is it? It's a guanine. It's a G. So you look at this and you say, okay, well, I've got this hydrogen bond. Was that a donor or an acceptor? Is this a donating hydrogen bond or accepting one? Accepting? Guess again. That there is an acceptor. Always. Okay? And this is a donor. So you look at this and you say, well, it's N7. Why can't this be an AT base pair? So this zinc finger binds GGG. That's what it does. Okay? GGG is its triplet. So the question you might ask is, well, what's going on here? That's just N7. There's no, I could have adenine here, right? So trick of crystallography. When they did this, they really can't see which atoms these are. This nitrogen, or this histidine side chain, this imidazole, can flip like this. Okay? When it flips, the nitrogen will go from here to here. Here's a funny thing. Here it acts as a donor of this hydrogen bond. If it flips over to here, and it's actually in this position, it acts as a donor to that. So in fact, probably what's really going on is it's doing this. It's sampling both. And obviously, if this was an NH2, that would be very bad. It would push it right off. So this guy really is specific for G. So here's a case where you have dynamic interaction. And you really don't know. Both of these are actually within hydrogen bond distance, which is too close for methylene. And so that's curious. Again, it's probably time average going like this. Where in the crystal, it's probably frozen half like this, maybe half like that. So GGG is its recognition sequence, and we can account for the specificity by the hydrogen bond pattern. So, again, opposite strand, same strand, same strand. So just because you recognize a GC base pair, it doesn't mean you recognize the G because I wrote it first. You can recognize either the G or the C to recognize a GC base pair. It just depends which strand you're on. Okay, so all of these guys that I've shown you have shape complementarity with DNA. You can see the surfaces interact quite nicely. Oh yeah, by the way, sorry, before I get there. One thing about the zinc finger, which is quite different from the other ones, this is not lying in the major groove. This guy's actually pointed into the major groove, like taking a cylinder and just sticking its end into the major groove. So it's not got the same sort of recognition helix type structure that the other ones have. It really is just kind of like this cylinder that you're sticking into the groove, and it happens to be coated with DNA binding residues. Okay, but all of these guys have some shape complementarity with DNA. In all cases, we saw backbone interactions that were ionic and hydrogen bonded with our typical expected residues. And we also saw examples of specific interactions in the groove, primarily in the cases I've shown you with alpha helices, but it isn't always that way. We also talk about beta strand in the minor groove. We also talk about alpha helices in the minor groove. I didn't show you pictures. And what I can tell you is there are definitely examples of beta strands in the major groove as well, but that's a lot rarer. There's something called a beta ribbon motif that does that. All right, so that is DNA binding proteins. Everything I think you need to know. Are there any questions? Okay, so this is what I'd like to do with the remainder of the class. For 15 minutes, if you care to, you may log on and do your eval. That would be great. If not, you can go, or you can come and talk to me, or you can come and pick up your exams. I will have an office hour today starting at 4:15 down in 3061 SLB. Anyway, I've enjoyed having you all in this class. I look forward to seeing you on Monday and also on Wednesday. That's not me clapping for me, that's me clapping for you guys.